Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Adaptive Immune Responses of Vertebrates, Part 3. Up till now, we have been discussing the innate, that was Part 1. We began looking at some of the components of the adaptive in Part 2. Well, here in Part 3, we're going to continue as we discuss the generation of what is referred to as a humoral response. Humoral means antibody-driven. Remember, antibodies, that Y-shaped thing that we talked about at the end of chapter, or Part 2 for this section of the chapter. The different classes of antibodies, remember I said, had their different roles. What starts the B cells being able to express and manufacture those antibodies is an interaction with a subset of the T cells. Okay, before the T cell can go and interact with the B cell, the T cell itself must be activated. T cells are activated when they are presented with an antigen by what they refer to as an APC, antigen presenting cell, APC. These are macrophages, dendritic cells, are chief two that will go and interact with the T cell. It's not just the macrophage or the dendritic cell comes up to the T cell. Hello, McFly, wake up, McFly. No. What the APC does is it comes and says, here is an antigen. Do you recognize it? That's it. That's what it does. The method, though, that the APC presents the antigen, that's a little more complex. Because remember, the macrophage is going to go and it's going to engulf. It's going to phagocytize and then digest via a lysosome into small peptide fragments, small fragments of glycoproteins, glycolipids. Most of those fragments it's going to continue to digest down and utilize the sugars, the amino acids itself as energy sources. But some of those, a very few, but some of those fragments are going to be loaded into an MHC. Remember, major histocompatibility complex. There's two classes, class 1, class 2. Class 1 is in every cell in your body. Class 2, well, that's going to be found in the APCs. The APC will load up that little fragment of a much larger antigen that, like I said, protein, lipoprotein, glycoprotein, or glycolipid. It will load it up into the cleft of the MHC. Remember, it has that cleft where things will be loaded in, and then it'll put that MHC on its surface. It then goes to B cells and T cells. Do you recognize? Do you recognize? Do you recognize? Do you recognize? Looking for any B cell or T cell that says, yes, I recognize that. The way it does this, as you see here, if you look here, you'll see that between the two cells, the one on the left is an APC, antigen presenting cell, okay, macrophage or dendritic cell. The one on the right is going to be a T cell. The T cell has coming on its surface a special type of receptor we call a T cell receptor. We'll talk more about those in just a slide or two. The macrophage or the dendritic cell, the APC, has on its surface the MHC. The MHC has loaded in its cleft something that is referred to as an epitope, which you see right there. What's an epitope, you say? Dr. Welsh, you haven't used that word yet. You can't use new words. It hurts our brain. I get it. Okay. If your immune system, remember, doesn't know a bacteria, it knows a protein, a glycoprotein, a glycolipid on the surface of that bacteria. What your B cells and what your T cells actually learn, actually recognize, actually will be activated by, is a small fragment of that protein, glycoprotein, or glycolipid. That small fragment of the much larger protein, that is what an epitope is. It's not the bacteria, it's not a protein on its surface, it's a small fragment of that protein. That fragment can be anywhere from 15 to 30 amino acids in length. It's a specific sequence of amino acids. It's a specific sequence of sugars and amino acids. Whatever. That's the epitope. That is actually what the adaptive cells in your immune system actually learn, what they will respond to. So when you look here between the two cells, you'll notice the one on the left, your left, yes, your left, the APC has on its surface the MHC with an epitope. Has a couple of other things sticking off that in this figure there that says co-stimulatory signals. What those are, don't worry. If you really must know, read that book that I linked on this chapter's page in Canvas, or take immunology with me. What we find happens when we've studied this interaction between an APC and a T cell. The APC comes in. On its surface is the MHC. In the cleft of the MHC is loaded an epitope. It comes up to a T cell. T cell has a receptor called the T cell receptor. Talk more about those in a slide or so. The MHC and the T cell receptor interact. And if the T cell receptor can recognize, interact, bind to that epitope, they will stay together. If the T cell receptor, once it interacts with the MHC, but cannot interact with the epitope, they pull apart. The cells go away from each other. But if the epitope inside the MHC cleft is something the T cell receptor can recognize, it's specific for MHC, T cell receptor, interact, then the epitope, which can now interact with the T cell receptor, stabilizes that interaction. But it's not completely stable. Okay, it's not going to hold forever, which is what the co-stimulatory signals are all about, which is what that CD4 thing you see looping over from the T cell to touch to the MHC. All of that stabilizes the interaction between the APC and the T cell long enough for the T cell to be activated. Once activated, that T cell is going to start producing many, many new things, because that activation by the APC upregulates, boom, upregulates certain genes expression. Some of those genes are going to be cytokines, specific class of cytokines known as interleukins. You notice here in this figure, you'll notice there's IL-1, IL-2, IL-4, 5, whatever. IL, interleukins, IL-1, IL-2, IL-3. Turns out each of these interleukins has a different effect on cells. So Here in this figure, it's showing that the T cell, once activated, will start manufacturing and releasing IL-1. IL-1, which can also be released by the macrophage, because once the macrophage interacts, remember that whole interaction and all those co-stimulations, the macrophage releases IL-1 also. Well, T cell releases IL-1 also. IL-1 will make all T cells that it comes in contact with start proliferating, specifically the one that's bound to the APC, the macrophage. So one T cell interacts with one macrophage. Macrophage, during that interaction, starts releasing IL-1, which makes the T cell get stimulated, gets activated, starts releasing its own IL-1, which, acts autocrine out and back. So that one activated T cell now starts dividing and dividing and dividing, starts also producing other cytokines. Each of those different cytokines are going to start having different effects, as you see here. Upregulating expression of different receptors, starts, upregulation of certain gene expression, certain, cell abilities now get turned on, others get turned off. What are the specific ones for which specific interleukins? Take immunology if you must really know. Just know for now, the macrophage starts the T cell, activates it, dumps on some cytokines, which causes the T cell to also start producing cytokines, start proliferating. Also, that you can have what we see here. You start over at the very end there, that big macrophage. That big macrophage, here the figure from the textbook that we're, the parasitology textbook, is showing a virus being phagocytized by the macrophage. The virus gets into a vacuole, because it's been engulfed. The virus then gets digested down. Some of those fragments of proteins, glycoproteins, glycolipids, get loaded into MHC. The macrophage then goes and finds a T cell, probably in the nearest lymph node. And it doesn't just go up to the first T cell and here you go, everything's good. No, it goes up to the nearest B cells and T cells. Do you recognize? Do you recognize? Do you recognize? Do you recognize? Until it can find one that has a T cell receptor. Here we're going to talk about those. A T cell receptor that recognizes that epitope. Remember part 2, antibodies. Talked about the fact that the antibodies at the end of the Ys had a variable region based on different sequences of amino acid giving different secondary, tertiary, quaternary structure and whatnot. And those different shapes, the different secondary, tertiary cause different rippling shapes, different that may or may not be able to interact and bind to the antigen. Well, it turns out T cells have T cell receptors that also have a variable region. They look quite a bit like the FAB fragment of an antibody, are not the same thing. They just look like immunoglobulin domains, constant area region, variable region. The variable region, because of the different amino acid sequences, will have a different shape at the ends, embedded within the variable. The different shapes can interact with different things, different epitopes. When the T cells leave the bone marrow, they've now hit that step where they've been released into the bloodstream. They're still naive. They haven't learned yet. No two T cells are exactly the same. Their variable regions are different. I mean, think about it. There's 20 different amino acids. These regions are going to be composed of dozens and dozens and dozens of amino acids, and you go through all the different permutations of 20 to the dozens and dozens power. That's how many different types of T cell receptors there could be. So what this macrophage is doing, if we go back to this figure here, is it's going one after another. Do you recognize? Do you recognize? Do you recognize? Do you recognize? Until it hits the one T cell that says, yes, I do. MHC, T cell receptor, boom. Epitope can bind to the T cell receptor, stabilizes. And as you see in this figure there, when you move over and you look at it, you'll notice that this means the T cell receptor, the T cell, here specifically a special type called a Th2, T helper cell type 2, will start releasing a long list of interleukins, those cytokines. The arrows, as you notice there, denote that it'll start going through its own maturation process. Starts going through its own maturation process and traveling to start going to B cells. Macrophage goes to a T cell, finds the correct T cell, activates it, then the T cell starts going around interviewing different B cells. Do you recognize this? Do you recognize this? Do you recognize this? Until that T cell, which has now been activated and matured, goes and finds the one B cell that has a B cell receptor, which, hey, once again, the B cell receptor looks exactly, almost like, but not quite, an FAB fragment of an immunoglobulin. When it finds the one that has the correct shape, because of the amino acid sequence in the variable region, T cell receptor, B cell receptor interact, they stable, co-stimulatory signals, and then you see that second set of arrows. That denotes the fact that now this B cell that has been activated by the Th2 cell is also being bathed in that nice long list of interleukins that the T cell started to produce. And well, that B cell will now start to go through a modified version of mitosis we call clonal expansion. Okay? One B cell becomes 2, those become 4, 8, 16, and so on and so on. Some of those daughter B cells will become the memory cells you see right there. The memory cells are B cells that kind of get set aside. We don't want you to do any work yet. You're here as backup in case we ever get exposed again. You're there, you're ready. Because if we've been exposed to this antigen, this epitope once, there's a good chance we're going to get exposed again at some point. The rest of those daughter B cells are going to differentiate and become what's referred to as plasma cells. Plasma cells are nothing more than antibody-producing machine cells. That's all they do. So that T cell receptor, back at the beginning there, on that last slide, okay? T cell receptor has to not only interact with the MHC, but it has to interact, the T cell receptor has to interact with the MHC and the epitope to be stable, okay? If the T cell receptor can't interact with the epitope, interacts with the MHC, but then it needs this additional little bit of interaction with the epitope to stabilize everything. That doesn't occur. APC, T cell don't play well with each other. They ignore each other and move on. Once it happens, co-stimulatory signals, lots and lots of cytokines will be produced. gene upregulation through the roof, and away things go. Because if you look at this figure right here, you'll notice in this figure that, when the first, what they call the primary immunization, that's first exposure, you're going to get a little bit of antibody produced. Because an APC is going to take that antigen from the vaccination. It's going to go to the different T cells, derecognize. That T cell is going to go find a B cell. Here you go. That takes days, as you notice. Takes up to almost a week or so. And then it's going to take, few days, almost a week beyond that, until you get maximal, that first hump, antibody production by plasma cells. Within a week, that quickly starts to decrease. You come in with the second vaccination, a second exposure. Instead of a week or so for B cells to get activated, that's within a day or so now. And look how much higher that second boom is. Real world. Okay, right now COVID's the thing. COVID vaccines, awesome. A lot of people ask, why does there have to be 2 shots? First shot, as you notice there, primary vaccination. Going to get a bit of an antibody response, a bit of a T cell response. Going to take a few weeks, but then it's going to quickly taper out. Come in with that second shot, because remember, most of the COVID vaccines now have 2 shots. 3 or 4 weeks later, you come in for the second one. That's the second, what they call the challenge. This really activates the immune system. Because notice, the y-axis there isn't linear. After the primary immunization, notice, goes up to 1 and then quickly drops back down. After the challenge, shoots up 1,000-fold. It's a logarithmic scale. 3-fold higher, 10 to the third higher. And when it starts to go back down, it doesn't go back down to less than 1. So what does that mean for us if you're walking around, you fly to somewhere new in the world, you get exposed to a new bacteria, a new virus, a new parasite. First exposure, it's going to take a week and a half for your immune system to learn it. Remember, learn it. For the correct T cell to be found, and that correct T cell to go find, interview a bunch of different B cells to find the correct B cell for you to start making antibodies. It takes about a week and a half. Within a week, all those plasma cells are going to start to go anergic. That means they're dying off. You still have the memory cells, though. You get re-exposed because you ate some food, got bit by the same insect vector a second time. A few weeks later, boom. Instead of having to wait a week and a half for your immune system to kick in maximally, now it's just a day or so, maybe even less than a day. All those memory B cells start going through clonal expansion, differentiate in, some of them will differentiate into plasma cells themselves. Off you go to the races.